Sir, we are live now. I request your permission to start the session, sir.
Yes, start now.
Thank you, sir. Hari Om, everyone. A warm and heartfelt welcome to the 37th session of our lecture series. A prestigious lecture series designed exclusively for the first year BMS and fourth year BMS students. We are delighted to have you all here today. But before we embark on this enlightening journey, let me take a moment to introduce you to Jignasa, a pan India platform driven by the student community of Bharata within the Ayush fraternity. Our motto is simple yet powerful. Learn Ayush to practice Ayush. We strive to empower Ayush students from campuses to communities, bridging the gap between students, policymakers, and administrative authorities. Jignasa serves as a unifying force connecting every aspect of Ayush, including students, academicians, practitioners, scientists, industrialists, and hospitals, all under one inclusive umbrella. We firmly believe in thinking globally while acting locally, working tirelessly over the past 2 decades to strengthen the Ayush education system and instill confidence among Ayush students. To achieve our goals, Jignasa has undertaken various impactful initiatives across the country, ranging from grassroots to international platforms, and some of our key initiatives include study circles, lecture series, free medical camps, social health status surveys, hands-on training workshops, conferences, interaction with eminent Vaidyas and academicians, brainstorming sessions, National Arogya Expo and International Conference, national level seminars, medicinal plant visits, pharmaceutical visits, agitations for high student community, private internship programs, interaction with traditional healers, online mock tests, social awareness campaign promoting various concepts of high system for societal well-being like Swarna Bindu Prashana Camp, Ashtashatottara Yoga, Group Surya Namaskara, and many more. Free healthcare assistance during natural calamities and accidents through local governing bodies were also provided by our organization. Yesterday's session was a testament to the overwhelming response and enthusiasm from students. With over 600 live participants and more than 25,000 views so far, we are humbled by your active engagement and dedication to learning. At Jignasa, we understand the financial challenges that many students face when it comes to accessing commercial study platforms. We firmly believe that education should be accessible to all regardless of their financial constraints. Hence, we are committed to providing a helping hand to every student fueled by the burning desire to learn. With the support of experts in the field, we offer you the opportunity to learn the subject with no financial burden. And all we ask for is your unwavering interest and passion for knowledge. Today, we are honored to have Dr. Akshay Kumar Cholin sir with you, BMS MD Ayurveda, Assistant Professor Kriya Sharira, Ayurveda Mahavidyalaya and Hospital, Hubli. Sir completed his UG from Ayurveda Mahavidyalaya and Hospital, Hubli in 2015 and PG from IM College of Ayurveda and Hospital, Asan. Sir, to list his achievements, Sir has organized many seminars and just a national level seminar on Kriya Sharira, state level seminar on Dravya Guna, national level seminar on Roganidana, and also delivered many guest lecture series, which include a guest lecture in Parul University, Vadodara, on the topic of understanding of Ruddikshaya Lakshana in day-to-day practice, guest lecture on biomedical waste management in induction program AMV Hubli, and many more. Sir, we are delighted to have you on this session today, and we extend our warmest welcome to you, sir, and to each and everyone watching this. We hope you find this helpful. Sir, now I request you to carry this session forward.
Thank you, Karthik. Good afternoon, everyone. Today I am presenting on exam orientation strategies on Kriya Sharira. So let us start with the Kriya Sharira paper. This Kriya Sharira paper is having 400 marks, and 200 marks is for theory and 200 marks is for practical. And in theory, again it is divided 100 marks each, that is paper 1 100 marks and paper 2 100 marks. And in case of practical, the practical examination carries 100 marks, and viva plus and internal assessment is for 100 marks. Before going to the topic proper, we'll have some guidelines to understand or to how to write exam and all. First, before going to the topic, I would like to clarify that this PPT is prepared for exam going students, those who are having basic knowledge on exam. And this PPT is prepared in keeping in mind that which are important points to read and which are very important points which are coming in the question paper, or which are going to come in question in exams. And how to write uh, how to write 5 marks question and how to write 10 marks question on same topic, that I am going to elaborate in this uh, presentation. Well, before going to exam, what you have to do, what the student who is going to appear for exam, what you have to do first, before the exams you have to by heart all the shlokas and keep it ready for exams. Because writing shlokas in the exam and saying and reciting shlokas in the practical viva, these carries more marks. And one more thing, you write the Sanskrit terminologies or Ayurveda terminologies in Sanskrit only or Devanagari lipi only. Don't write with English. It is not wrong, but the impression will be bad. The presentation or the way of presentation will be very bad. It won't look good, and the evaluator will not find it is good. So keeping in mind all these things, by heart shlokas are very important. They also carries marks. If you write shlokas, you will get good marks. If you don't write shloka, you will not get good marks, right? Next is read all the by this time you have to complete the syllabus at least for 3 times. Because coming next week or another 15 days we will be having exam. By this time you have to complete the whole syllabus of Kriya Sharira at least by 3 times, so that you can get good marks. Next, go through the syllabus copy and don't leave any of the syllabus points unread. Because the question paper blueprint and the syllabus copy is designed in such a way that every point should be covered in question paper. Every syllabus point should come in the question paper in such a way that it is framed, so that you should cover all the syllabus points and don't leave any of the syllabus points unread. Because at the end you will pay for this. Next, prepare the topics before appearing to the exam. Prepare the topics for same topic for 5 marks also and for 10 marks also. So how you have to prepare, that will see in the further ppts, further presentation. So first, before going to exam only, you prepare a same topic. For example, vata dosha. For example, this vata dosha can be written for 10 marks also, for 5 marks also. So how to write, how which and all points should be covered in 5 marks, which and all points should be added in 10 marks, that you have to decide before going to exam only. If you decide this thing in the exam or while writing the exam, it will consume lot of your time. So before that only you have to decide how much you have to write, how much you have to write for 10 marks, how much you have to write for 5 marks. This is the thing. And before writing or during the exam, while writing the exam, first read the question once or twice. It won't consume a lot of time reading the question. Next, by reading once or twice, you can understand the questions very easily. So understand the question. Don't write what you know, write what it is asked in the question. Okay, don't write what you know, write what it is asked in the question paper. Right? Understand the marks division. For example, uh, explain vata and its types in detail for 5 marks. You just understand the question and marks division of the marks based on this question. For explain vata, for vata it is half marks, and for its types it is half marks, which means 5, 2.5, 2.5, it is divided. That division you have to understand. It comes by experience and it comes by the perfection on the subject or grip on the subject. That will be there. And manage time. So time management is usually a big headache for students. It is very easy when you decide the what amount of or if you decide the points to be written on the same question, on the question. then you can manage the time, and it is very important and it is very crucial one. Next is draw diagrams. See, drawing diagrams is very important, and this also carries marks. And the drawing diagrams is not the diagram should not be a perfect one. It is near to perfect is okay. Okay, so don't focus it on perfection of the diagram, but it is near to the structure of the diagram, that is okay. Organ or a diagram, it is okay. Next, and wherever the diagram is necessary, there you draw a diagram. Okay, next point is make a points in the answer paper and uh, answer the question. Because many of the students, what they do is they write from they start in the first top of the first page, and they directly ends in the last line of the same page. It is very wrong. It is not a wrong, but it is the way of presentation is very wrong. So try to make a points, try to make a paragraphs, leave some space or leave some lines in between the two paragraph, and make sure that each paragraph consists of minimum maximum 4 to 5 five lines. And draw flow charts, draw columns, and time and tables. You have to represent in such a way that the types and all you can make a flow charts, right? And if something is having a 10 cells and it's a secretions, that for that you can make some table or columns. That makes the evaluator to easy understanding, evaluator to easily understand what you have written. Okay, the easier understanding of the evaluator is very important. And next last point is, don't leave any of the questions unanswered. Because if you leave any of the questions unanswered, then the chances of passing is very difficult. So don't leave any of the questions, and answer all the questions. Manage time properly. Make points and paragraphs. Draw diagrams, flow charts, and tables should be there in your answers. So that makes the good presentation of your answers. Next, and before starting my presentation, I would like to share one more thing. Here in this new syllabus, the answer booklet will be like, first you have to write long answers, that is 10 marks, and there will be four long answers, and each long answer will be having 6 pages. Okay, each long answer will be having 6 pages. That is, this pattern is according to RGUHS Bangalore, which are the colleges coming under RGUHS Bangalore. For them it is applicable. Okay, first question, first the long answers will be having 6 pages. Each long answer will be having 6 pages. For example, question number 1 will be having page number 1 to page number 6. Question number 2, 7 to 12, like that it will be there. And next attempt short answers, that is 5 marks. 8 5 marks question will be there in the questions. And for each question, 3 pages is given. For example, question number 5, it is having page number 25 to 27. Next, question number 6, 28 to 30, like this it will be arranged in the answer booklet. And the last, MCQ, there are 20 MCQs, and for 10 MCQ, 1 page is given. And the next 10 MCQ will be written in the next page. That is, 13 to 22 MCQs will be in 49 page number, and 23 to 32 MCQ will be on 50th page. This is the thing. Next, we'll move on to the topic proper, that is paper 1, Kriya Sharira paper 1, part A. Here I have prepared my present PPT based on the marks allotted. So here, topic 1, topic 2, topic 3, all three together the allotted marks is 8 marks. So topic 1 is Shareera, topic 2 is basic principles of Ayurveda, topic 3 is Tridosha. For all these three topics, three syllabus points, 8 marks is allotted, which means 3 MCQs and one short answer will be there. One 5 marks question will be there from these three topics. So understand the marks allocation, and then you have to study. First, we'll move on to Shareera. First syllabus point. What you have to write, what you have to read in this Shareera topic. Here you have to read definition, synonyms of Shareera and synonyms of Kriya. These synonyms of Shareera and Kriya are asked in MCQs, and many a times the MCQs are usually prepared in such a way that there will be four answers or four options for the each MCQ, and these four answer in that four this thing four options. First, the correct answer and the next answer will be very similar. Correct answer and the next option will be very similar, and the third option will be related to that topic, and fourth one will be completely not related, in such a way that the MCQs are designed. So don't put the wrong answer, put a right answer for the MCQ, and for that you need, if you want to write the correct answer, for that you need to have a proper knowledge, and without any, you should not have any confusion regarding that topic. That is the thing. So in definition, Shiriyate Aninaiti Sharira, that you have to read synonyms of Sharira and synonyms of Kriya. And next is difference between Sharira and Sharira. Sharira and Sharira, that has to be studied, because it is very important, and it is many a times this Sharira and Sharira questions are asked in MCQ. So you so don't take risk in MCQs. Next is Sharira dosha and Manasa dosha. In case of Sharira dosha, there are three doshas, Vata, Pitta, Kapha, that the introduction of all these three things you have to read. And in case of Mano dosha, Raja and Tama, the introduction of all these two things you have to read. And Sattva is the Prakashaka, and it is considered as Guna of the Mana. So all all the doshas of Sharira dosha and Mana doshas are you have to read. Next is mutual relationship between Triguna and Tridosha. So here the how you have to explain the mutual relationship between Triguna and Tridosha with the Panchamahabhuta is you make a table. First, in first column you write a dosha, in second column Triguna, and in third column you write Mahabhuta. And Vata, which is having a Trigunas like Sattva and Raja, and Mahabhuta like Akash and Vayu. In such a way that you have to explain, you have to draw a table, and then you have to explain. Only drawing a table is not sufficient, what I feel is. So you have to explain it completely, and you have to draw the table. That is the thing. Next syllabus point is basic principles of Ayurveda. In that there are two points, that is dosha dhatu malam moolam hi shariram. The explanation of this dosha dhatu malam moolam hi shariram, the you have to explain, you have to read the things like the dosha concept, dhatu concept, and mala concept, and understand the how our body is made up of. In order to understand all these things, this concept is given. And the second concept is Srotas. See, I have displayed one chart on a table, which is having a name of Srotas and Moolasthana according to different samhitas, like Charaka Samhita, Moolasthana of Charaka Samhita, Moolasthana of Shushruta Samhita, and Astanga Sangra. So these three samhitas has been referred, because the each and every samhita explains different Moolasthana, which is differing slightly, and that can be asked in Viva. That can be asked in Viva also, that can be asked in MCQ also. So it is very important. So this can be asked in 5 marks also. So if it is asked for 5 marks, what you have to write? So first is basic, first is definition of shrotas. Shrotamsi kalu parinamam apadyamananam dhatunam abhivani bhavanti ayanarthena. That shloka you have to write. And next, you have to write the types of shrotas. Next, types bahya and abhyantara mukha shrotas, that you have to write. Next, the samanya lakshana, samanya dushti nidhana, that you have to write. Next, mula of the shrotas according to different samhitas, that you have to write. If it is asked only for charaka samhita, then it fits for 5 marks. If it is asked all the three acharyas, then it fits for 10 marks. But here no question of 10 marks, because this topic is allotted only 5 marks for 5 marks, allotted only 5 marks. So any of the samhita can be asked in this. Next is Tridosha Siddhanta. In this, relationship between Ritu and Dosha and Rasa, and biological rhythm of Tridosha on the basis of day, night, age, season, and food intake. That is explained. That is asked here. If the question is asked on Rutu and Dosha, then you have to write Chaya Prakopa Prasamani Vayur Grishmadishu Trishu. That shloka you have to write, and you have to explain. Or if the question is asked on Dosha and Rasa, then Tattadnya Marudam Gnanti Trayotiktadayah Kafaha. That shloka you have to write, and um, you have to establish the relationship between dosha and rasa. Which rasa causes the ruddhi of which dosha, and which rasa suppresses the ruddhi, or which rasa does the kshinata of which dosha. That you have to explain in detail. Next is biological rhythm. For this, there is a shloka, वयो अहोरात्रि भुक्तानां ते अन्त्यमध्यादिकः क्रमात्. That shloka you have to explain, and there it is clearly mentioned that how different doshas are, how vata pitta kapha are dominant in uh, different parts of the day, different parts of the night, different parts of the age, or different ages, and different seasons, and food intake after, before, and that also it is mentioned. Better, whenever you are writing these kind of answers, you make a table. For example, for biological rhythm of day, night, age, season, and food, you make a table which is having, in first column which is having doshas, and in second column day, in third column night, in third column age, in next column season, next food intake. In which time of the, in which part of the day, the uh, which doshas are dominant, that you have to write in that chart, or in that table. That that improves your presentation. That is the good way of presenting this answer. Okay. Next is role of doshas in formation of prakriti of an individual. So there are different doshas. There are three doshas which influences the formation of prakriti during the uh, conception. uh, in that, you have to write the first you have to give an introduction and a definition. You have to write shukra shonitam samyoge yo bhavet dosha utkataha. That shloka you have to write. If possible, you write another shloka, prakriti nama janma marnantara kale, that is from rasa vaisheshika, that you have to write, you can write. And then you come for doshas influencing the um, formation of prakriti. And there also you can explain factors affecting the prakriti, that is intrauterine and extrauterine factors, like shukra shonita prakriti, kala garbhashaya prakriti, mathura ahara vihara prakriti, mahabhuta vikar prakriti. And in case of extrauterine factors like tatra prakriti, प्रकृति जाति प्रसक्त कुल प्रसक्त देश प्रसक्त देशानुपाति सॉरी देशानुपातिनी कालानुपातिनी वयो अनुपातिनी प्रत्यात्मनीयता in such things can be explained in the role of dosha forming the prakriti of an individual. Okay, don't write only shlokas, write its explanation along with the shloka. Next is role of doshas in maintenance of health. See, this topic can be asked in MCQ, that is multiple choice. Samadosha Samagnesha, that shloka can be taken as a reference, and by that you can say that Sama Avastha of a dosha leads to health, and equilibrium, the inequilibrium state of the dosha leads to disease. And last point is Prakruta and Vikruta doshas. Prakruta dosha is the one which is in normal stage, or which is in normalcy, and it maintains the normal functions of the body and in the normal way. But in case of Vikruta doshas, these are the one which is in vitiated state. So they are responsible for the formation of disease. This is about the tridosha topic point. So these, so next we'll move on to the questions, probable questions of multiple choice. You cannot guess the probable questions in case of multiple choice, but what I can say is one question from Shareera can be asked, one question from basic principles of Ayurveda can be asked, and one question from Tridosha can be asked in this MCQ. Next, 5 marks questions in Shareera. It is first is define Shareera, synonyms of term Shareera, and a difference between Shareera and Shareera. This can be answered for 5 marks. Okay. Next is description of Shareera dosha and Manas dosha. So this can be, so the here you have to understand the question and marks allocation. Here Shareera dosha, for Shareera dosha 2.5 marks, and Mana dosha 2.5 marks is allotted. And Shareera dosha, Tridosha Vata Pitta Kapha can be introductory way you have to explain. And Mana dosha, the Sattva Rajatama can be explained in this, and even with the Panchamahabhuta. Next is 5 marks questions on basic principles of Ayurveda. The only one topic which can be asked in this syllabus point is Stotas. Here, this question is asked for 5 marks because no question of 10 marks. Define define the Shrotas, types of Shrotas according to different Samhitas. This can be asked, or define Shrotas, Shrotodushti Karanas, and uh, and uh, and explain Shrotas according to Charaka Samhita. That also be asked, or Sushruta Samhita, that can be also asked. This is 5 marks question. Next, on Tridosha, describe interrelation between Rutu Dosha Rasa and explain biological rhythms of Tridosha on the basis of day, night, age, and food intake, season and food intake. Previously told, there are 3 shlokas you can write, and you have to explain those three shlokas, and you try to represent that answer in the tabular form or table form, so that the evaluator can easily understand the answer. Because you are in Karnataka, in RGHS, the question the answer papers are scanned and they are evaluated in computers by the evaluators, right? So it is very difficult to see a computer for longer duration. If your answer is well represented in the form of tables, then it is very easy to understand for the evaluator also. So that gives you a good impression on your paper. Okay. Next is role of dosha in the formation of prakriti of an individual and factors influencing prakriti. So the role of doshas are vata pitta kapha during the formation of embryo. How which dosha is predominant, that prakriti will be formed. And there are different factors of influencing prakrutis, that is in intrauterine and extrauterine factors, that is shukrashonita prakriti, kala garbhashaya prakriti, maturahara vihara prakriti, and mahabhuta prakriti. And other factors and extrauterine factors like um, जाति प्रसक्त कुल प्रसक्त दोषानुपातिनी कालानुपातिनी वयोनुपातिनी प्रत्यात्मनीयता. So all these can be written in this for 5 marks. Okay, and whenever you are writing answer, you try to highlight the specific important points, and highlight the subheadings, and try to maintain the numbering. Okay, that is very important. Next we'll move on to next topic number 4, 5, 6, 7, 8, that is vata pitta kapha and dosha vruddhikshaya, that is vata pitta kapha vruddhikshaya and kriya kala. So here one 10 marks question will be question is there, and two 5 marks question, so it becomes 20, and 6 from the mcq. First point, that is fourth point, vata dosha is having two mcqs, and pitta 1, kapha 1, dosha vruddhikshaya 1, and kriya kala 1, so it becomes 26. This is the marks allocation for these 5 points, that is vata dosha, pitta dosha, kapha dosha, dosha vruddhikshaya, and kriya kala. We'll see one by one how it should be how it can be studied and how it can be written in the exams. What are the probable questions, how we have to answer these probable questions, right? First is vata dosha. So here you have to write the nirupti, you have to write the utpatti first, next nirukti, next location samanya, general location, that is general location, that is sthana of the vata dosha, and properties, that is guna of vata dosha, next karma of vata dosha, and the types of vata, that is prana udana samana vyana apana. These are to be studied in the heading of vata dosha. And if the question is asked for 5 marks, you write uh, up to the types of vata, which means you have to write utpatti, nirukti, the term nirukti, and location, that is sthana, then guna, then karma of vata, and you have to name the 5 types of vata. That is sufficient for 5 marks. Okay, there are for derivation, there is a shloka, that is tatra vagati gandanayoho iti dhatu. And for location you can write pakvashaya kati sakti shlotrasti, that shloka you can write guna tatra ruksha lagu shitakara sukshmaschalo anila, that. And karma, there are 4 to 5 shlokas for karma. You decide how how much marks it is, for how many marks it is asked. Then after that deciding, you have to write the shlokas. Simply don't write 4 5 shlokas, or don't write big shlokas for 5 marks, or by that doing that you can waste your time. So you are going to waste your time, so don't do that. If you are well enough to write in a short period of time, you can write, but think twice and then you start writing the shlokas. For function, that is karma, utsaha uchvasa nishvasa chesta vega pravartani, that shloka you can write. And in case of 10 marks, for 10 marks, if the same question is asked for 10 marks, same topic is asked for 10 marks, then along with the till the function you have to explain. Along with this, you have to explain the prana udana samana apana vyana apana individual types of vata doshas, with the specific location, that is sthana, and properties, and its function. Each and every type of vata dosha is having 2211 line shloka, 22 line shlokas, that you have to by heart, and that is needed for that 10 marks questions. Along with ruddhikshaya lakshanas can be asked there also. Next, same thing with the pitta dosha, utpatti, nirukti, the sthana of the pitta dosha, uh, gunas of the pitta dosha, and karma of the pitta dosha, types of pitta dosha, that is pachaka, ranjaka, alochaka, brajaka, sadhaka, and these things can be written in the pitta dosha. One more thing I forgot to explain, I forgot to tell in the vata dosha. Shwasana prakriya in Ayurveda, it is there in the syllabus, and for that you have to write Nabhistha Pranapavana, that shloka, that is from Sharangadhara Purvakanda 5th chapter. That shloka you have to write. That is very important, and this is the reference where the Shwasana karma is explained in Ayurveda. That you have to write. Okay, that can be asked for 5 marks, but that can be asked for 5 marks. That is the thing. Next, one more thing is there in vata dosha, that is physiology of speech in Ayurveda, vak utpatti. Okay, that is from Paniniya Shiksha. It is explained in that Samhita, and you can refer that. You can refer that, but it is in desire to know. We won't expect the questions for 5 marks. Okay, next is in vata only. I am continuing in vata only. In vata, rasarakta samvana is with the help, it is explained with the help of vyana and samana vayu. That is, the shloka goes like this: vyanina rasadaturhi vikshepojita karmanaha yugapat sarvato ajasram vikshipate sada. This can be asked in this shloka can be asked in multiple choice, because there are two terms, there are two words which are important in these shlokas, that is yugapat and ajasram. So those can be asked in sorry, so those things can be asked in MCQ, and the meaning of yugapat, the meaning of ajasram, so those things can be asked in uh, MCQ point of view. Okay, that is vata dosha. Next, pitta dosha. So these things can be explained in pitta dosha for 5 marks. That is utpatti, nirukti, uh, sthana, guna, karma, for 5 marks. If it is asked for 10 marks, then you explain the individual types of uh, 5 types of pitta, and the shlokas of pachaka pitta, ranjaka pitta, alochaka pitta, those things can be written, and along with that you have to write the vruddhikshaya. Then also that answer fits for the 10 marks. Right, same thing with the kapha: utpatti, nirukti, sthana, guna, karma, and 5 types of kapha are explained here. You have to study all these things, and you have to study all these things. Okay, this is applicable for kapha also: vata, pitta, kapha. On any of these 3 topics, you can expect the 10 marks question. And also the next chapter is, next one is Kriya Kala. This, on this also we can expect a 10 mark question. First, before going to Kriya Kala, we will see, we'll look into the uh, syllabus point, that is dosha vruddhikshaya. Here, the here what you have to study is etiological factors responsible for dosha vruddhi and dosha kshaya. That you have to study, that you have to read. There is a shloka, that shloka you have to by heart for all the three doshas, for Ruddhi and for Kshaya. Next, dosha Ruddhi and Kshaya Lakshana Shloka you have to write, which is very important. Usually it is asked in all the Vaivas. Okay, this can be asked for 5 marks. Etiological factors, Ruddhi Kshaya Karanas of Vata dosha. Like this, it can be asked, and Ruddhi Lakshanas of Vata dosha, Kshaya Lakshanas of Vata dosha, or combining two of the Ruddhi, one of the Ruddhi and one of the Kshaya doshas, of the Kshaya Lakshanas of the dosha. That can also be asked in this chapter. Next is Kriya Kala. So when we come to the Kriya Kala, what is the concept? First, you have to understand the concept of Kriya Kala. Kriya Kala means Kriya means the action, Kala means the time. It is the ideal time for the for the disease to prevent, or you can plan the treatment. That is the Kriya Kala. And here you have to explain the types of Kriya Kala. That is Vyadhi Kriya Kala, and they are six in number. That is Shad Kriya Kala, that is also called as Shad Kriya Kala, and Rutu Kriya Kala. For Vyadhi Kriya Kala, you have to write the shloka like Sanchaya cha prakopam cha prasaram sthana samshayam vyakti beda cha yo veti doshanam sa bhavet bishop. So that shloka is must and should. And for Rutu Kriya Kala, Chaya Prakopa Prasamani Vayo Grishmadishu Trishu, that shloka you have to write. And then you explain the Shat Kriya Kalas or Shat Kriya Kalas. Okay, the stages of six Kriya Kala. If it is asked for Vyadhi Kriya Kala, then you have to explain all the Kriya Kala. If it is asked for Rutu Kriya Kala, it is very easy. You write the shloka and make a chart, make a table, and write the Vata Pitta Kapha, which Rutu it is. It becomes the Ruddhi, and which Rutu it becomes Siksha. That you have to write. Okay, this is about the Kriya Kala. We can expect a 10 marks question or 5 marks question from this. Next, we'll move on to the discussion on probable questions. As I told, MCQ cannot be predicted, uh, but we can. It is clearly mentioned that in the syllabus point, Vata dosha, on the Vata dosha, two MCQ can be asked, and Pitta, Kapha, Vruddhikshaya of dosha, and Kriya Kala, one MCQ questions can be asked here. Next is probable 5 mark questions. Here, two questions from the 5 marks can be asked. As I told in the while explaining the Vata, it is, I have cleared that how much you have to write for 5 marks, how much you have to write for 10 marks. So in case of Vata, in case of 5 marks, if the question is Vata dosha, utpatti, nirukti, general location, that is sthana, guna, karma, then it is sufficient for 5 marks. You have to write all the shlokas with its explanation. Okay, that you have to write. This is for 5 marks. You can expect question like this also: 5 types of Vata, or 5 types of Pitta, or 5 types of Kapha. What is 5 types of, what are the types of Kapha, with their specific location, specific properties, guna, karma, sthana? Those can be asked for 5 marks also. So here you can divide the marks like each type can carry 1 marks, which includes shloka and its explanation. Don't write only shlokas, write its explanation also. Okay, next, any of the Ruddhi Kshaya can be asked in 5 marks, and Kriya Kala in detail with example can be asked for 5 marks. Kriya Kala, Vyadhi Kriya Kala, with one example can be asked for 5 marks. And probable 10 marks question here, one 10-mark question on these 5 topics can be expected. Uh, here it may be on Vata or Pitta or Kapha, uh, full Utpatti, Nirukti, Sthana, Guna, Karma, and its types, along with Ruddhi and Kshaya Lakshanas can be asked here. So you first, before writing only, you decide how much you have to write, what and all things you have to write, and what is asked in the question, that you decide, that you understand, then start writing the questions, start writing the answers. Okay, next, we'll move on to next further topics, that is topic number 5 to 12. For these topics, 26 marks are allotted. That is Prakriti, Ahara, Agni, and Aharapaka. For these 4 topics, 26 marks has been allotted, which means 1 10 marks, 2 5 marks questions can be asked, and the rest of the multiple choice questions can be asked here. So let's move on to the topic. They have in Prakruti. This Prakruti, first you write the derivation. You write the derivation. Pra and Kruti means to create. Uh, that is, you write that you have to write. Next definition, Shukra Shonit Samyoge, that you have to write, and factors influencing a Prakruti, that is intrauterine and extrauterine factors, you have to write, and individual Prakruti Lakshanas can be written here. Vata Prakruti, if it is Deha Prakruti, you write Vata Prakruti. Sorry, before going to the individual types, you write the classification of, you read the classification of Prakruti. Prakruti can be classified into Sharira Prakruti or Deha Prakruti and Manasa Prakruti. Deha Prakruti is having three types: Vataja, Pitaja, Kaphaja Prakruti, and Manasa Prakruti is also having three types: Satvika, Rajasika, Tamasika. And in case of Satvika, it is again divided into 7 characters: Brahma, Indra, Varuna, Kubera, Gandharva, Yama, Arsha. And Rajasika are classified into 6 characters: Asura, Sarpa, Shakuna, Rakshasa, Pishacha, and Preta. And in case of Tamasa, it is having 3 characters like Pashva, Matsya, and Vanaspatya. So all the individual characters, if it is asked, then you have to write. Don't write for only 5 marks. Okay, you write whenever it is needed. You decide the amount of what you have to write, the quantity what you have to write. Then you have to explain the individual, each and every point of the individual things, individual characters should be explained here. That is, Brahma, what are the characters of Brahma? Indra, what are the characters of Indra? That you have to mention, that you have to explain, okay, while writing the Manasa Prakruti for 5 marks, or only Manasa Prakruti for 5 marks or 10 marks. Okay, next we'll move on to Manasa Prakriti is over. Next we'll move on to Ahara. Here definition, classification, and significance of Ahara is Ahara we have to read. In that definition, Aharyate Galad Adunyate, that definition you have to read. And classification, in classification there are many classifications explained in the mentioned in the books. First is classification according to origin, that is Stavara and Jangama. Stavara is having 4 types: Vanaspatya, Vanaspatya, Vanaspati, Viruddha, and Aushadha. And in Jangama, it is again 4 types: Jarayuja, Andayuja, Svedayuja, and Udbija. These are the classification. Another classification is on the effect: Hitakara and Ahitakara Ahara. And another classification, uses, based on the uses: Peya Lepa, Peya Lehya, Bhojya, and Bhaksha. That is based on the use, on which form we are taking the food. That is the thing. And based on rasa, shad rasas, based on shad rasas, the ahara is classified. Based on guna, vimshati guna, ahara is classified. Based on panchamahabhuta, ahara is again classified. This classification should be mentioned while writing the, while if it is asked classify ahara, then you have to explain all these classifications. Next, importance of ahara and its significance. For this you have to write the shloka pranaha pranabhutanam annam uh, loko abhidhavati. That shloka, that is from Charaka Sutra 28th chapter. That shloka you have to write. Sorry, that shloka you have to write, and you have to explain the shloka. Next, ahara vidhi vidhana. Okay, ahara vidhi vidhana, sorry, this definition, classification, and significance of ahara, that can be asked for 5 marks, right? Next point is ahara vidhi vidhana. There are some guidelines for taking the food. Those are 13 guidelines has been explained in the Ayurveda. Those are Ushna, Snigdha, Matravat, Jirna, Virya Viruddha. Like that, it is, they have mentioned 13 types of ahara vidhi vidhanas. Okay, don't write blindly, don't write you have to take ushna ahara, you have to take snigdha ahara, you have to take matravat, you have to take food after the previous food is digested. Don't write only that thing. Writing that that much is very important, but only writing that sentence is not important. In that, you have to write the significance of taking ushna ahara, why we are going to take ushna ahara, that you have to mention. Then only it will carry a good impression on your answer paper. Then only you will get a good number of marks. Okay, so while writing ahara vidhi vidhanas, you have to explain all 13 ahara vidhi vidhanas with its significance or with its importance. Why we have to take ushna only, why we have to take ahara without laughing, why we have to take ahara without um, taking without talking. So such things have been explained in this chapter. Okay, next is asta ahara vidhi visheshayatana. So these are the eight ashta ahara vidhi vidhanas. Eight ahara vidhi vidhanas are visheshayatanas has been explained: prakriti, karna, samyoga, rashi, desha, kala, upyog, samstha, upyogta. For each and everything there is a shloka that you have to write. And uh, and you and at the last you write, you try to explain the importance of all these things: ahara vidhi visheshayatana. Okay, next point of this ahara topic is ahara parinamakara bhavas. There are some there are some uh, mm, there are some things which with the help of this the digestion takes place. That is ushma, vayu, kleda, sneha, kala, samyoga. So all these things are helping in digestion, and that is ahara parinama. Parinama means digestion. And so these are the characters, these are the things which helps in ahara parinama. Those things has to be written with the written with its significance or importance. And at last uh, agni is the topic. So in this you write the definition of agni, synonyms of agni, and types of agni. There are 14 types of agni. And try to explain the difference between pitta and agni. Try to explain difference between pitta and agni. You have to write, you have to read, and you have to write. You make a better, you make a tabular form or a table form, and you have to explain the agni. So types, in that types: jatragni, bhutagni, and dhatwagni. And after this type classification of agni, where mandagni, tikshnagni, and vishamagni, that has to be explained in detail. Okay, this is this answer should be written, these all points should be written in case of agni. Next, ahara paka. Ahara paka means the digestion of ahara. Annam adana karmam tu prana kosta prakarshati. This line, this shloka should be explained, this shloka should be written, and this 6 line shloka should be written in this ahara paka. Okay, that is the digestion of ahara. Next is avastha paka. Avastha paka is nothing but the food which is in the middle of the digestion, which is not the digestion is here, the digestion is not completely digested. There are three types of avastha paka: madhura avastha paka, amla avastha paka, and katu avastha paka. So for each and every avastha paka there is a shloka. For example, madhura avastha paka shloka is अन्नस्य भुक्तमात्रस्य षड्रसस्य प्रपाकतः मधुराध्यात् कफो भावात् फेन भूतम् उदीर्यते. That is the shloka of madhura avastha. So like this it should be written and it should be explained in detail. And which are the doshas getting generated, those things has to be stressed here. And those things should be written in the while writing the avastha. नेक्स्ट आम्लावस्थापाक परन्तु पच्यमानस्य विदग्धस्य आम्लभावतः आशयम् आशयश्च अवमानस्य पित्तम् अच्छम् उदीर्यते. दिस इज द श्लोका फॉर आम्लावस्थापाकम्. नेक्स्ट कटु पक्वाशयं तु प्राप्तस्य शोष्यमाणस्य वनिनां परिपीडितपक्वस्य वायुस्यात् कटुभावतः. That is the shloka and you have to explain the shloka in case of avastapaka. This can be asked for 5 marks. Okay, avastapaka can be asked for 5 marks. Next, after the avastapaka, we have to go to the will see the vipaka, that is nistapaka. So they again these there are three nistapakas or the three vipakas has been mentioned. Madura and Amlarasa is getting converted into is its vipaka is Madura vipaka. Madura and Lavana rasa is Madura vipaka. Amla rasa is Amla vipaka. Katutikta kashaya will be in Katu vipaka. They are going to Katu vipaka. Vipaka means the food after the digestion. It can be see the effect of this vipaka can be seen on the dosha, on the dosha, on the dhatu, and on the mala. Vipakaha karmanishtayaha. So how you are going to judge the vipaka means by its action on the different parts of the body, different entities of the body. So that is vipaka. And here one more thing can be added here. Here you can differentiate the avastapaka and vipaka, avastapaka and nistapaka. Those differences can be written with the help of table, with the help of table that should be written here. And some of the points you should not miss the miss here is anna mahashrotas should not be missed, that should be read. And grahani is the one where pitta is where the pitta is secreted. That point of view, it is very important. Grahani, that should be read. And pittadara kala, pittadara kala also be studied. And costa is very important. And in this costa, definition of costa, and importance of definition of costa, types of costa, and its significance of costa, and at the last costa pariksha should be written here. That is in case of costa. Next, we'll move on to the MCQ. Probably MCQ questions on this. First, Prakriti will be having two MCQ questions, and one MCQ on Ahara, one from Agni, and two MCQ questions from Ahara Paka. So there are so many questions for 5 marks. These can be asked, that is, write Nirukti, synonym of Prakriti, classification and characteristics, features of each kind of Deha Prakriti in detail. So you have to write the Nirukti, synonym of the Prakriti, classification and characteristic features of each kind of a Deha Prakriti. That is Vata, Pitaja, Kafaja. Those three things, the Sharangadara Shloka should be written here. If you can write a Charaka's Shloka, then it is well and good. Minimum thing is Sharangadara Shloka is needed for Vata, Pitta, Kapha Prakritis. Okay, that is for 5 marks. Next is define Prakriti and classify Prakriti and explain Manasa Prakriti in detail. As I previously told, definition of Prakriti and classification of Prakriti, that is Shareera Prakriti and Manasa Prakriti, and explain Manasa Prakriti in detail. And you have to explain the different types of Manasa Prakritis, that is Satvika, Rajasika and Tamasika, and each and every kaya should be explained here. Characteristics of each and every kaya should be explained here. That is Brahma, Indra, Varuna, Kubera, Gandharva, Yama, Arsha. Those and Rajasa and Tamasika Prakriti should be explained here. Characteristics. Okay. Next is explain intrauterine and extrauterine factors influencing Deha Prakriti and classification and characteristics of each kind of Prakriti. Same thing of Deha Prakriti and intrauterine and extrauterine Prakriti is asked here. First, you understand the question and then answer. Okay. Next, definition, classification, and significance of Ahara. See, this is clearly discussed in the Ahara topic, that is clearly explained, that is definition and classification. Different classifications has been mentioned, and the importance and significance of the ahara has to be written. For that, one sloka is there that I have told, and that sloka has to be written here. And explain, second, next question is explain ahara parinamakara bhavas, or ahara vidhi visheshayatana, or ahara vidhi vidhana in detail. See, here you can expect ahara parinamakara bhava, or ahara vidhi visheshayatana, or ahara vidhi vidhana for 5 marks. Anything can be asked for 5 marks here. Next, definition of Agni, synonym, location, properties, functions, classification of Agni in detail. Here, this is also mentioned. This is also explained. First, define Agni and its synonyms, location where it is located, functions, and classification of Agni um, has to be explained here. Explain Annavastrotas and their Moola, and role of Grahani and Pittadarakala. This also can be asked. Explain Avastapaka. Next, explain Nistapaka and its classification. Explain genesis of Vata, Pitta, Kapha during the Ahara Paka process. This is nothing but the um, this is nothing but the avastha paka only you have to write, where in which stages different doshas are, in which and all stages the different doshas are getting arise, and those things to be explained here. And next is define the term costa and classification of costa and characteristics of each type of the costa should be written here also here. So this is about the 5 marks. Then examples of 10 marks question. Write nirukti, synonym of prakriti, classification and characteristics of characteristic features of each kind of a deha prakriti and manasa prakriti. Previously I told, if the deha prakriti and manasa prakriti both are asked, then it is for 10 marks. You write it in detail. You have to write in detail for 10 marks. All the characteristics of each and every prakriti, each and every type of prakriti should be explained here. Okay, define classification and significance of ahara in detail, and explain ahara parinamakara bhavas, or any of the these things can be asked, that is ahara parinamakara bhava, ahara vidhi visheshayatana, or ahara vidhi vidhana can be asked here. That is for 10 marks. Next is define and classification of avastapaka, nistapaka. Classification of avastapaka and nistapaka, and difference between avastapaka and nistapaka you have to write. First, define classification of avastapaka and define classification of nistapaka, and then write the difference between the avastapaka and nistapaka. For this is for 10 marks, so that carries the 10 marks. So this completes the paper 1 part A, that is for 60 marks. Next we'll move on to paper 2 part B, that is for 40 marks, that is modern. Here first three syllabus points have been allotted with 23 marks, that is physiology of homeostasis, respiratory system, gastrointestinal system for 23 marks, which means one 10 mark question, two 5 marks question, and three MCQs can be asked in this topic, right? So keeping in mind that how much you have to read, how what should be read in these topics, which are the important points, which are the important topics, that you have to keep it in mind. First we'll move on to physiology of homeostasis. Here, definition and mechanism of maintenance of homeostasis. First point is this one. So, definition of homeostasis is maintenance of constant internal environment of the body is called as homeostasis. And here you have to read role of various systems of the body in homeostasis. For example, pH of the body. So the 7.4 pH is maintained in our body. If it is not maintained, the metabolic process will not be carried out. The body temperature, same way, in the same way, body temperature. Nutrition and its supply, it should be supplied properly. Oxygen supply, hormones, water and electrolyte balance, it should be maintained, it should be balanced. And blood acts as a medium for all the above said functions. And skeletal muscles, this skeletal muscles, how homeostasis in skeletal muscles, this skeletal muscles helps to move around and search for the food. That that in turns, it helps in procuring the nutrition which is required for the body in order to maintain its normalcy. That is the thing. And next is, and last one is CNS and ANS. These are the two nervous systems which plays the master key role in this maintaining the homeostasis. Okay, this is the role of various systems of the body in maintaining the homeostasis. Next, this thing should be read. Okay, next this thing should be studied. Next is components of homeostasis. So what are the components of homeostasis? First, sensor, transmitter, controlling center, and effector. These are the components. This is to be studied. And mechanism of action of homeostasis. So this is the very important point of very important point in case of 5 marks and in case of 10 marks also. So how the homeostasis works in our body? It is whenever there is a deviation in the system, that is corrected by means of feedback mechanism. So there are two feedback mechanisms have been explained. One is negative feedback mechanism, another one is positive feedback mechanism. So by seeing this name, what students do is negative negative feedback mechanism, they'll think that it decreases the deviation. And positive feedback mechanism, it increases deviation, but it is not so. Negative feedback mechanism is a one which reacts in such a way that it arrests the change or reverse the direction of a change. Okay, what is negative feedback mechanism? It is the one that it reacts in such a way that it arrests the change or reverse the direction of change. Example, ADH, water balance in our body. Water balance, if the water concentration of the water is decreases, then the through feedback negative feedback mechanism, the person is the water balance is maintained by thirst mechanism and other different mechanism, ADH mechanisms. And it maintains the normal water level in the body. If the water balance, if the water concentration is more, then the by stopping the thirst, and by ADH mechanism, there will be a lot of urine is formed, and that is the excess water in our body is eliminated in the form of urine. That is done. That is negative feedback mechanism. Here the correction of uh, deviation, which may be in the which is in the opposite direction, takes place. If it is increasing, it decreases. If it is decreasing, it increases. So don't get confused by looking its name, by seeing its name. Okay, what about positive feedback mechanism? For positive feedback mechanism, it is a system, it is the system reacts in such a way that it increases the intensity of the change in the same direction. That is, the example is clotting or clotting and parturition. Okay, these two examples can be given here. So the in case of whenever the mechanism of action of homeostasis is asked, you have to write the positive and negative feedback mechanism. Positive You have to understand the positive and negative feedback mechanism properly. Don't get confused by its name. It is not, this name is not given for the nominal sake. Okay, it won't be decreases for a negative feedback mechanism, it won't be increases for for positive feedback mechanism, it is true, but for negative feedback mechanism it is not so. Okay, next syllabus point is cell physiology and membrane physiology. In this, what you have to study is definition of cell, definition of tissue, definition of organ, definition of system, that you have to study. And structure of cell. Cell is made up of mainly three structures, that is cell membrane, cytoplasm, and nucleus. Cell membrane is protein layer. It is made up of protein layer. It is enveloping the cell body, and it is semi-permeable membrane. It is composed of 55% of proteins and 40% of lipids and 5% of carbohydrates. Okay, that you have to study. And structure of cell membrane, there are three different models has been explained in the uh, explained for this cell structure of cell membrane, that is Davison model and unit membrane model and fluid mosaic model. So these three models have been explained for the structure of cell membrane. And wherever there is a diagram is necessary, there you have to draw a diagram. For example, structure of cell, where the you can draw a cell membrane, you can draw a cell with the nucleus and cell membrane and cell organelles, that you can draw, right? So please draw diagrams wherever it is necessary. Next is functions of cell membrane. There are six functions of cell membrane which is explained in the books. So those things has to be studied. Next comes to the next we'll move on to the cytoplasm, where all the cell organelles are present in this, in the cytoplasm. There are the we can classify these cell membrane cell organelles in two forms, that is with limiting membrane and without limiting membrane. With limiting membrane cell organelles are endoplasmic reticulum, Golgi bodies, lysosomes, peroxisomes, centrioles, vesicles, mitochondria, nucleus. And without limiting membrane is ribosome and cytoskeletons. For in order to explain all these things, you have to make 2 points. That is, you have to explain these with the two templates. That is, structure of the particular organelle and its function with its diagram. In this way, you can explain this. For example, mitochondria. What is the structure? What is the function of mitochondria? That you have to write. Okay. In this only two templates, you can write this. You can explain all these cell organelles. That is, one is structure, second one is function and its diagram. Okay. Next, you have to read the nucleus. You have to read the genes. You have to read DNA, RNA, and DNA replication, RNA replication. Those things has to be studied in this topic. Okay. Next is transportation of various substances across the cell membrane. Here, the essential substances like nutrients, water, electrolytes, and other things are transported across the cell membrane. So it can be classified into two types: active transportation and passive transportation. In active transportation, the energy is required, and in passive transportation, there is no need of energy in passive. Again, these active and passive are divided into several types. For example, passive sorry uh, for example, uh, passive transportation is again divided into simple diffusion, facilitated diffusion. This simple diffusion is again divided into simple diffusion through lipid layer, simple diffusion to protein layer, and carrier mediated diffusion. So all these points can be should be answered, or all these points should be explained while explaining passive transportation mechanism. Next, active transportation mechanism. It is divided into two types, that is primary and secondary, and there are special type of active transportation mechanism are explained for macromolecules which are um, bigger than these channels or these pathways. So they are endocytosis, exocytosis, transcytosis. Okay, these three are special type of active transport mechanism. And there are channels has been explained. These are the pores in the cell membrane, and they are called as channels. And they are made up of proteins, and they are specific to ions, so they are also called as ion channels. And these channels are classified into two types: gated channels and ungated channels. Gated channels are again divided into three types, that is voltage gated channels, ligand gated channels, mechanically gated channels. So these all points should be covered in this channels. Okay. Next we'll move on to applied physiology of this transportation and these channels. Here, abnormalities of sodium and potassium pump has to be explained. Diseases of channels and etc can be diseases of channels can be explained here. So this carries this so all these points should comes under the transportation of substance across cell membrane. Okay. Next syllabus point is resting membrane potential and action potential. So resting, what is resting membrane potential? First you have to study the definition. It is defined as the electrical potential difference across the cell membrane under resting condition. So there are different tissues are having different resting membrane potential. For example, human muscles, skeletal muscles are having minus 90 millivolts. In this way, neuron is having different value, and cardiac muscles, smooth muscles, erythrocytes, different tissues are having different values of this voltage. Okay. And the next, development and maintenance of this resting membrane potential, that has to be studied. That is mainly dependent upon the potassium ions, mainly dependent upon the potassium ions. Next, ionic basis of resting membrane potential has to be studied here. And there are two factors responsible for this imbalance. There will be imbalance in the ionic basis of resting membrane potential, because uh, because uh, because uh, in presence of the in case of sodium and potassium ions, two potassium ions are taken in with the exchange of three sodium ions. So there will be net loss of one positively charged ion inside the cell. So that produces the that develops the resting membrane potential. There will be one net loss of positively charged ion, so that leads to the development of resting membrane potential. There are factors responsible for the resting membrane potential are sodium potassium pump and selective permeability of the cell membrane, that is channels for major ions and leak channels. In case of um, in case of heart, in case of uh, uh, nodes of the heart, there are leaky channels of sodium. Because of these leaky channels, continually there will be generation of impulses, continually there will be uh, production of action potential, and that leads to generation of impulse and propagation of impulse takes place. Because of that, the heart beats, that heart contracts. That is the thing. So those things are comes under the resting membrane potential definition. uh, Different tissues having different resting membrane potential, development and maintenance of resting membrane potential, and ionic basis of resting membrane potential, factors responsible for resting membrane potential has to be studied under the heading of resting membrane potential. And in case of action potential, The definition should be studied, that is series of electrical changes that occurs in the membrane potential when cells are stimulated. There are it has got two phases. One is depolarization, another one is repolarization. Depolarization means it is negatively charged, negatively the cell is charged positively inside the cell, and outside it is negative. And repolarization is nothing but the initial stage of resting membrane potential, that is inside negative and outside positive. That is the action potential. So these points are covered under action potential. Next, acid base balance. This acid base balance is studied under different systems, that is in blood, and in respiratory system, and in renal system. This acid base balance is studied, and even this water and electrolyte balance also same thing. Here our body is made up of 60 to 70% of the water, and there are two sources of water that has to be studied, that is exogenous source and endogenous source. Exogenous means from externally we are what we are providing, the what water is providing to the body. And endogenous means because of the food metabolism there will be production of water, and that leads to the that becomes the endogenous source of water. And the significance of body fluid, there are five significance of body fluid are explained, that is homeostasis, transportation, metabolism, metabolic reaction, texture of the tissue, and temperature. All these things are the comes under the significance of body fluid. Next, components of body fluid are intracellular. These, and again these are divided into different headings, so those things are studied here. Okay, next is electrolytes. What is definition of electrolyte? The substance which is dissolved in a solution and that dissociates into ions, that is positive ion and negative ion. That is that is called as electrolyte. There are two types. Classification of electrolytes is two types: cations and anions. Cations are positively charged and anions are negatively charged. The factors regulating electrolytes are homeostasis and biochemical factors. Uh, so these all points are studied under the heading of electrolyte, water balance, and acid base balance. Okay, next point in the syllabus point is study of basic components of food. So there are different components of food. Food is a basic requirement for the living being, and there are different constituents of the food. For example, there are five basic components, that is carbohydrates, proteins, fats, vitamins, and minerals. For all these there is a template. In these templates only it has to be written for easier explanation, that is definition, source, and examples. If it is having any hypo and hyper these things, then that also can be included here. So for example, carbohydrates. What is the definition of carbohydrates? That has to be written. Source of carbohydrates and examples for the carbohydrates, that has to be written here. So those all things should be read, studied for the components of food. Next is respiratory system. So in the respiratory system, first syllabus point is functional anatomy of respiratory system. Here the structural things which are through which the air moves in and out, those things are considered as the um, functional anatomy of respiratory system. For example, nose, pharynx, larynx, trachea, and bronchi. These things has to be studied here. Pleura. What is this pleura? Pleura is a bilayer serous membrane which covers the lungs, and it has cavity uh, that cavity is between the two layers of pleura, and that cavity is filled by fluid. That fluid is called as pleural fluid. There is a there is some significance for that fluid, because it prevents the friction by lubricating it, and it creates a negative pressure. It creates a negative pressure in the on the lungs. Okay, that is the pleural fluid, intrapleural fluid. And next you have to study the tracheobronchial tree. What is this tracheobronchial tree? It is formed by the two things, one is trachea and one is bronchi. So that has to be studied, and the trachea is bifurcated into left and right primary bronchiole. So, and again it is further divided into secondary bronchiole, tertiary bronchiole. When right in case of right lung, it has 10 tertiary bronchiole, and in case of left it has 8 tertiary bronchiole. It is because why 8, why in case of left why 8 and right why 10? It is because of the heart, which is accommodated in the left side, so that is consuming some space. For that there is the tertiary bronchioles are less in number. And uh, and uh, again it is divided into several times and it becomes bronchiole. And next again it is converted into terminal bronchiole when the diameter is 1 mm. And next at last the respiratory bronchiole, when it is called as respiratory bronchiole when the diameter is 0.5 mm. When the diameter is 0.5 mm, that is the terminal bronchiole. So all these things should be studied under the heading of functional anatomy of respiratory system. And here there are some functions of the respiratory tract. The main function of respiratory tract is to conduct or carry the air from external atmosphere to the the this thing alveoli or alveoli, and from alveoli uh, to the atmosphere. That is the main function of this respiratory system. But apart from these there are different functions. They are called as non-respiratory functions of respiratory tract has been explained. They are olfaction, vocalization, filtration of air, defense mechanism, maintenance of water balance, body temperature regulation, acid base balance, anticoagulant function, secretion of ACE, that is angiotensin converting enzyme, synthesis of hormones. So all these functions are explained in non-respiratory functions of respiratory tract. These this non-respiratory functions of respiratory tract is very important for the point of 5 marks question. And a respiratory protective reflex. So there are different reflexes are there are three reflexes has been mentioned in case of respiratory protective reflex, because these reflexes protects the respiratory tract from the irritation or from the damage. For example, cough reflex, it is a one of the respiratory protective reflex. And it is seen only when there is a irritation in the mucous membrane of the respiratory tract. Sneezing reflex, it is seen whenever there is a irritation in the irritation in the mucous membrane of nasal cavity. So these are the respiratory protective reflex. Deglutition reflex, so this deglutition reflex protects the respiratory tract from entering the food into the respiratory tract. Okay, by closing the epiglottis, the food by closing the epiglottis, the food directly goes into the um, esophagus. That is the deglutition reflex. And uh, and the next point is pulmonary circulation, pulmonary artery. That includes circulation to the pulmonary artery, bronchial artery, and physiological shunt. And characters of pulmonary blood vessels, that has to be studied in this topic only. And regulation of pulmonary circulation should be studied in the functional anatomy of respiratory tract. This functional anatomy of respiratory tract can be asked for 10 marks. Okay, and next we'll move on to Next we'll move on to mechanism of next we'll move on to ventilation and mechanism of respiration. Ventilation means it is defined as the air which is moving in and out in the particular duration of time, that is called as ventilation. And mechanism of respiration, it occurs in two ways, that is inspiration and expiration. Inspiration is active and expiration is passive, and there are different muscles which are taking place in which are working for the respiration, that is inspiratory muscles and expiratory muscles. So that has to be studied here. And movements of thoracic cage. So whenever there is inspiration and expiration, it should be there is this should be coordinated with the thoracic cage enlargement, and enlargement of the thoracic cage, and this thing. So this movement of thoracic cage is uh, mainly depends upon the four structures, that is thoracic lid, upper coastal series, lower coastal series, and diaphragm. So the movement of thoracic lid, the movement of upper coastal series, the movement of lower coastal series, and the movement of diaphragm are very important in the movement of thoracic cage. They increases the horizontal and longitudinal length of the lungs. Okay. And in this, one more point should be studied, that is while studying the upper coastal series and lower coastal series, there is a mechanism mentioned, that is pump handle mechanism and bucket handle mechanism. These are like a similes quoted for the ribs, movement of ribs. So based on that, you can understand how the ribs are moving in our body while inspirating and expirating. And the next is movement of lungs. Movement of lungs. In movement of lungs, whenever there is a enlargement of thoracic cage, the lungs expands, and whenever there is a contraction of thoracic cage, the lungs collapses, and there will be the collapsing tendency of lungs is there, and that has to be studied. There are some factors which are causing the collapsing tendency of the lungs. And one more thing, the surfactant word, that the terminology is there, that is the surfactant that has to be studied. These all things are studied under the heading of mechanism of respiration and ventilation. Mainly mechanism of respiration. Next is mm, next is exchange and transportation of gases. Here two words are there, one is exchange and another one is transportation. In case of exchange of gases, the partial pressure of the oxygen in the atmosphere and partial pressure of the oxygen in the alveoli, because of there will be change in the partial pressure of the oxygen in the atmosphere and alveoli. Because of that, the oxygen diffuses into the alveoli. From there, it diffuses into the blood, because of the pressure difference in the oxygen, partial pressure of oxygen. And see, in the same way, in the blood, the partial pressure of oxygen is more, and in the tissue, partial pressure of oxygen is less. Because of that, it diffuses into the uh, tissues. In the same way, in the reverse way, the carbon dioxide moves. Exchange of carbon dioxide takes place. That is, from the tissue to blood, the partial pressure of carbon dioxide is um, less when compared to tissue. It moves into the blood, it diffuses into the blood. In the same way, blood to alveoli, alveoli to atmosphere. In this way, it comes out of the body. This is the mechanism of exchange of gases, that is carbon dioxide and oxygen. These are the two things. And uh, and in case of transportation of gases, here two gases comes, that is one is oxygen and second one is carbon dioxide. Oxygen is transported in two forms, one is dissolved form and another one is combination with hb. Here, oxygen carrying capacity of hemoglobin is 1.3 ml per gram. That you have to remember, and it should be studied, and it is explained. Okay, that is transportation of in the under the heading of transportation of oxygen. In case of transportation of carbon dioxide, it is transported in four forms. The carbon dioxide is transported in four forms. One is dissolved form, it forms 7% of the total transportation. And the second one is carbonic acid form, it is very negligible. And in third one is bicarbonate ion, it forms the 63% of the total transportation of carbon dioxide. And last one is carbamino compound, it forms the 30% of the total transportation. In all these three forms, the transportation of carbon dioxide takes place. And it should be explained in detail when it is asked for 5 marks. If it is asked for 10 marks, both the carbon dioxide and oxygen both should be written here. Next is regulation of this respiration. Regulation of respiration, that is neural and chemical control of respiration. In case of neural mechanism of regulation, respiratory centers are involving in this, and efferent nerves are involving in this. And in case of respiratory centers, there are two respiratory centers, medullary centers and pontine centers. And these two are having different neurons, that is dorsal group of neurons and ventral group of neurons in case of medullary center, and apneustic and pneumotaxic centers in pontine center. So these are the nucleuses uh, which controls the respiration. And in case of chemical regulation of respiration, the chemoreceptors plays an important role in regulating the respiration. There are two types of chemoreceptors. One is central chemoreceptors, and another one is peripheral chemoreceptors. The central chemoreceptors are situated in brain, and peripheral chemoreceptors are situated in aorta and carotid sinuses. So whenever there is a change in the chemical property of the blood, that will be sensed by the central and peripheral chemoreceptors, uh, and that will be sent for the respiratory centers. So this is about the regulation of respiratory centers, that is neural and chemical control of respiration. Next is artificial respiration. Before going to this point, we'll move on to pulmonary function test. Last point, that is pulmonary function test. Introduction to pulmonary function test. This is to assess the functional status of the lungs with the help of spirometer. There are two types of pulmonary function test: a static lung function test and dynamic lung function test. So lung these are the two types. And and in this topic only, there are two things has been explained, that is lung volumes and lung capacities. In lung volumes, there are four types: tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. And in lung capacities, inspiratory capacity, vital capacity, functional residual capacity, total lung capacity. These are the four lung capacities which are explained under the pulmonary function test, right? So this is the uh, these all things should be studied under the pulmonary function test. And the next is there are some terminologies should be studied. They are artificial respiration, they are apnea, hyperventilation, hypoventilation, hypoxia, oxygen toxicity, hypercapnia, hypocapnia, asphyxia, dyspnea, cyanosis. These are important when it comes to the MCQ. These are important when it comes to the MCQ. And the next point is artificial respiration. So artificial respiration is done artificially when it is not in capability to when it is not in capable to run its own run its own. that is artificial respiration. It has two classifications, one is manual and another one is mechanical. Manual is again divided into manual is having two methods. One is mouth to mouth method, another one is back press method. And in case of mechanical method, it has got two methods. One is drinker's method, another one is ventilation method. So these are the two methods of mechanical artificial respiration. Sorry. So these all points should be covered under the heading of artificial respiration. Next is physiology of gastrointestinal system. So, in the first point is functional anatomy of gastrointestinal tract. In that, you have to study the functional anatomy of digestive system, organs, primary and secondary organs. Primary organs like mouth, pharynx, esophagus, or accessory organs like teeth, salivary gland, exocrine part of pancreas, liver, etc., etc. These all are studied. And next important point to be studied under this heading is walls of GIT. Gastrointestinal tract wall is made up of four layer. One is muscular layer, sorry, mucus layer, submucous layer, muscular layer, and last one is serous layer. Here there are three types of muscles which are present in the muscular layer. It is They are inner oblique, middle circular, and outer longitudinal. This inner oblique muscles layer of muscle is absent in case of intestine. Okay, only two muscles of muscular layer is present in the intestine. And the nerve supply to the GIT, that is enteric nervous system, has to be studied. That is orbax plexus, Meissner's plexus has to be studied in this heading only. Next, we'll move on to the individual organs like salivary gland. So these there are different glands and different secretions in the GIT. For example, in the mouth there will be secretion that secretion is called as saliva, that is secreted by salivary glands. In the stomach there is a secretion called as gastric secretion, which is secreted by gastric glands, in the pancreas. In the pancreas there is a secretion uh, that is uh, that is called as pancreatic secretion. That is it is transported into the it is transported into the small intestine in the second part of the duodenum. So there are different types of secretions in the GIT. So how to study all these things? I have made some template, some points or some templates. So based on that, you have to study all these things. For example, salivary gland, you just start with the introduction, what is salivary gland, and situation. Second point is situation, where it is situated. And if it is having any types, then you explain the classification of salivary gland or types of salivary gland, and what is the weight of salivary gland, what is the quantity of secretion, which what is the duct through which the secretion is secreted. Next, composition of secretion. Next, functions of secretion. Next, functions of salivary gland. Next, regulation, sorry, next is regulation of this salivary secretion. And last is applied physiology of the gland. So through all these headings, this gland has to be studied, right? In the same way, for example, another example I would like to quote here is gastric glands in the stomach. So situation in the stomach, what is the quantity of the secretion, that you have to study, that you have to write. Composition of gastric secretion, that composition has to be studied. What is the function, that has to be studied. And regulation, that has to be written. And at last, applied physiology of all these things has to be done or read. In the same way, all the glands of the GIT has to be studied. from starting from the mouth to large intestine. Okay, so all these things are studied under these headings. Okay, next is phases of gastric secretion. It is very important because there are three phases of gastric secretion. One is cephalic phase, gastric phase, intestinal phase. And one more important point is mechanism of secretion of hydrochloric acid in the stomach. That has to be studied. Okay, in the same how the salivary gland is explained, in the same way pancreas to be explained, liver and gallbladder should be explained or studied. In the same way, small intestine, large intestine should be studied. Okay, so next, we'll move on to the topic that is movements of GIT from the mouth till the large intestine. Whatever the movements are taking place in the GIT that are explained here: mastication, deglutition, and in case of deglutition, there are three stages of deglutition: oral phase, pharyngeal phase, and esophageal phase. These are the three stages of deglutition. And movements of stomach. In movements of stomach, there are three types of movements: hunger contraction, uh, receptive relaxation, and peristalsis. These are the different types of movements which are seen in the stomach. Movements of small intestine and movements of large intestine are to be studied. Defecation reflex and gastro defecation reflex. In this defecation reflex, mechanism of defecation reflex and defecation pathway should be elaborated. Next, gastric gastrointestinal hormones has to be is the topic, where 15 hormones of gastrointestinal system are explained. These hormones are studied under three classes or three columns. One is hormone, one is secretion, another one is action. So you prepare one table which is having three columns. One side you write the name of the hormone, you another side in the middle you write the source or which cells from it is secreted, and next is what is the action of this hormone. So there are 15 hormones are explained. I don't go much detail about these things, already it is too late. Next is digestion of carbohydrates, digestion of proteins, digestion of lipids, and absorption of carbohydrates, absorption of proteins, and absorption of lipids, and metabolism of carbohydrates, proteins, and lipids has to be studied under this heading, in this subject only. So when we come to the multiple choice, the probable question or examples of questions, there will be one multiple choice from one each topic. That is physiology of homeostasis, one topic, one multiple choice, sorry, and physiology of respiratory system, one question, and gastrointestinal system, one question. In case of 5 marks question, there are two 5 marks questions are asked in this in these three topics. Questions may be what is homeostasis and explain mechanism of homeostasis. In that you have to write the negative and positive feedback mechanism also, and explain positive and negative feedback mechanism with example. That is very important. Here the marks distribution is differs from the first question. And what is cell and explain its organelles in detail. Okay, that is the where cell what definition of cell carries 1 marks, and organelles carries 4 marks. And you can keep 1 mark for we can keep 1 mark for diagram also, 3 marks and plus 1 marks. So that is how the marks distribution is done. And you have to plan your answer before answering this way, in this way only. What is transportation across cell membrane and explain active transport mechanism. Next question is explain passive transport mechanism in detail. What is resting membrane potential and explain action potential. Write note on basic components of food. These are the 5 marks questions. Respir- uh, Next is define respiration and explain structure of respiratory system in detail. Uh, And non-respiratory functions of respiratory system can be asked for 5 marks. Respiratory protective reflex in detail, all the three reflexes has to be explained here. Don't name them, don't name them, but you have to explain all these three things in detail. Pulmonary circulation and its regulation has to be studied, is asked, and its regulation is asked. Pulmonary circulation is 2.5, and regulation is 2.5 marks. And explain, next question is explain movements of thoracic cage. Here the movements of thoracic cage is depends upon the different structures of mm, structures. They are thoracic lid, upper coastal series, lower coastal series, diaphragm, all these things are there. Those things has to be mentioned. What is, next question, what is pulmonary function test, and explain lung volumes in detail. This five in that 5 marks, 2.5 for lung function, and lung volumes for 5 marks. And lung capacities in detail, that is for 5 marks. Explain exchange of oxygen in detail, or carbon dioxide in detail. So here you here there may be a question of exchange on oxygen or exchange on carbon dioxide. Both can be asked. Here exchange is asked. Remember, don't get confused with exchange and transportation of oxygen. Whatever it may be oxygen or it may be carbon dioxide. Exchange is different and transportation is different. Exchange till the uh, exchange is nothing but the diffusion. Transportation is carrying. Okay, so so understand the question and then answer. And next question is chemical regulation of respiration in detail. So chemical regulation is nothing but the uh, it is done by chemoreceptors, and that has to be answered here. Neural regulation, this is new this is nothing but the respiratory centers and efferent and efferent nerve fibers. In respiratory centers, different respiratory centers has been explained, that has to be written here. And artificial respiration, two types, that two types has to be explained in detail. The first you have to define, and then you have to explain its classification. Next is what is digestion, and explain structure of digestive system, that is functional anatomy of digestive system. Next is salivary glands in detail for 5 marks. Enteric nervous system and its function. Applied physiology of salivary gland. Explain stomach and gastric secretions in detail. Amount of gastric secretion, the contents, composition of gastric secretion, that has to be written here. Phases of gastric secretion, that is 3 cephalic, those things has to be written here. uh, mechanism of secretion of hydrochloric acid has to be is in that how the hydrochloric acid is released in the stomach, and how bicarbonate ions are secreted in the pancreatic secretion, that has to be explained here. And mechanism, explain the pancreas in detail. Explain pancreas in detail. Explain liver in detail. I have given a template. Based on that template, you have to explain all these things up to the large intestine. Next, explain deglutition or movements of stomach in detail. It can be asked for deglutition or it can be asked for stomach. Next is defecation reflex. That also can be answered. What are the gastrointestinal hormones, and explain any two hormones in detail. So you list out the 15 hormones, gastrointestinal hormones, and in that you explain any two of the hormones in detail. Next is applied physiology of liver, applied physiology of pancreas, applied physiology of gallbladder, small intestine, large intestine. Okay, these are also asked for 5 marks, and that has to be answered. Uh, Next is digestion of carbohydrates, proteins, and lipids. Any one can be asked. And next is absorption, metabolism, same. Digestion, absorption, metabolism of each and every thing can be asked here, that is carbohydrate, protein and lipids. Next is example for 10 marks questions. Here you can differentiate how 5 marks questions are framed and how 10 marks questions are framed. Here see what is homeostasis, mechanism of homeostasis, and explain positive negative feedback mechanism with one example. Here everything about the homeostasis you have to write. In case of 5 marks, if it is asked for 5 marks, there will be no need of explanation of example, or only homeostasis and its mechanism is asked, or only positive and negative feedback mechanisms are asked. So that is how the questions are, that is how you have to modify your answer for 10 marks for 5 marks. Next is what is transportation across cell membrane, and explain active transport mechanism and passive transport mechanism in detail. Next, mechanism of respiration in detail, transportation of oxygen and carbon dioxide, exchange of gases in detail, that is both oxygen and carbon dioxide you have to write. Regulation of respiration in detail, here neural and chemical regulation both you have to write. And what is pulmonary function test, and explain lung volumes and lung capacities, both are asked. That is what, how you have to change the answer. So you have to write the pulmonary function test, the types of pulmonary function test, and lung volumes, lung capacities in detail you have to explain. Next is functional anatomy of GIT, and explain next, explain liver and gallbladder in detail. What are gastrointestinal hormones, and explain in detail. Explain movements of GIT from mouth to defecation, that has to be explained. Digestion of carbohydrates, proteins or lipids, anything can be asked. And digestion and metabolism of carbohydrates, lipids and proteins can be asked for 10 marks. Next, we'll move on to last two topics of the paper 1 part B, that is nervous system and endocrine system. Uh, for these two topics, 17 marks has been allotted. One 10 marks question can be asked here, and one 5 marks question can be asked here, and two MCQs can be asked in these two topics, that is nervous system 1 MCQ and endocrine 1 MCQ. So which and all the topics coming under one second, which and all topics coming under these nervous system. Here uh, general introduction to nervous system and neurons. Here you have to give the introduction to the nervous system. What are the types of what are the classification of nervous system, central nervous system, peripheral nervous system, that has to be studied. In central nervous system, brain, spinal cord, those things has to be studied. Peripheral nervous system, in case of peripheral nervous system, somatic, it is classified into, it is divided into somatic and autonomic nervous system. In autonomic nervous system, sympathetic and parasympathetic divisions are explained. And next is neuron. Structure and functional unit of the nervous system is called as neuron, and its classification depending, there are different three types of classification has been mentioned, uh, depending upon the number of poles, bipolar, unipolar, multipolar, that classification has been mentioned. And depending upon the functions, motor function, sensory function, that is that another classification. Depending upon the length of the axon, Golgi type 1, type 2, these are the two uh, classifications of, another classification of neurons. And the next is structure of neuron. Structure of neuron consists of three parts: nerve body, that is nerve cell body, dendrite and axon. Here you have to study the myelin sheet also. Next is Classification of nerve fiber. The classification of nerve fiber, there are six classifications. One is depending upon the structure, distribution, origin, function, secretion of neurotransmitter, diameter and conduction of the impulse. Those things are being explained, that has to be studied. And next is propagation of nerve fibers, sorry, properties of nerve fibers. In that, excitability, there are seven properties of nerve fibers has been mentioned: excitability, conductivity, refractive period, summation, adaptation, um, infatigability, all or none law. Those things are to be studied in case of properties of nerve fibers. Next is functional. Uh, Next is functional anatomy of spinal cord. In that, you have to write the you have to mention the you have to read the situation of the spinal cord, where it is situated, what is the covering, meninges are the covering, what is the length, 45 centimeter is the length of the spinal cord, what are the enlargements, cervical and lumbar, these two are the enlargements, segments 31 segments has been made, and from each segment one pair of nerves are uh, emerged from the each segment. And gray matter of the spinal cord, white matter of the spinal cord should be studied here. And diseases of spinal cord should be um, studied, that is applied physiology of spinal cord, multiple sclerosis and disc prolapse, disc bulge. Those things are to be covered in this topic. And when it comes to spinal cord and spinal nerves, you have to study one more thing, cranial nerves. Okay, those cranial nerves are very important, that also been asked in the for asked, that can be asked for 5 marks. Next is sensory and motor nervous system. In that, um, uh, somatosensation and special sensation are the sensory nerves, and uh, uh, sensory nervous system. And it has two types, it has three types, and sensory pathway is mentioned, that has to be studied. And different parts, and that has to be studied in the same way. Motor nervous system has to be studied. Next is different parts of the brain, and different parts of the brain should be studied. For example, thalamus. Thalamus, where it is situated, situation, nucleuses, functions, applied physiology. In these headings only this thalamus should be studied, and while writing the exam you have to follow same pattern for writing the exam, so that saves your time and you can present it in better way. And hypothalamus also same thing, situation, nucleus, functions and applied physiology. And next point is learning and memory. What is learning? It is process by which new information is acquired. Two types, non-associative and associative learning. So what are these non-associative? Those things should be studied, okay, in case of learning. And in case of memory, definition, what is the definition of memory, that has to be studied, that is ability to recollect or recall the past experience or in past information, that is memory. And there are different types, there are four types of memory: anatomical basis of memory, physiological basis of memory, chemical basis of memory, and consolidated memory. And another classification of memory is short term and long term, explicit and implicit memory. So and applied physiology of this memory should be explained here, that is amnesia, dementia, Alzheimer's, etc. are to be covered, or are to be covered in this topic in case of speech and learning and memory. In case of speech, here the definition, mechanism of speech, mechanism of speech, development of uh, mechanism of speech, development of speech, those things should be studied. And one second, those things should be studied here. uh, And development of speech, there are two stages of development of speech. And area of speech, Wernicke's area, Broca's area, motor area, those things should be studied. And applied aspects of speech is aphasia, dysphonia, and stammering. These all are to be covered in the speech topic. Next topic is sleep and dreams. In this sleep definition, sleep requirement, that is for different age group people it is different, that should be mentioned. And physiological changes, there are during the sleep there are 10 physiological changes explained in the textbooks. Those 10 physiological changes should be written in the sleep physiological changes of sleep. And there are two types of sleep: REM sleep and NREM sleep, that is rapid eye movement sleep and non rapid eye movement sleep. There are different stages of sleep according to EEG pattern. Sorry, there are different stages has been explained according to EEG pattern. There are four stages of sleep based on EEG pattern has been mentioned, that has to be studied. And mechanism of sleep and center of sleep has to be studied. Next is applied physiology of sleep, insomnia, hypersomnia. Those things should be studied in the in case of sleep. Next is special senses. In special senses, functional anatomy, for example, uh, eye, special senses, in special sense, eye, functional anatomy of eye should be written, should be studied. Structure of rods and cones should be studied. And visual pathway. In the same way, uh, structure of ear, external middle ear, inner ear, and auditory pathway should be explained, or it should be studied. In case of sensation of taste, taste buds, situation, types, structure of taste buds, pathway of taste, that should be explained here. That should be written. And in case of sense of smell, olfactory pathway should be written. Next, last point is physiology of temperature regulation, body temperature regulation. Here, body temperature, variation of the body temperature varies according to the surroundings and according to the metabolism. Heat gain, when heat is gained when there is a increase in the metabolism, muscular activity, and hormonal, because of the influence of hormone and because of the shivering, the heat is gained. When heat is lost because of the radiation and all, the heat is lost. That those things should be studied. Mechanism of regulation of body temperature, that should be studied. Applied physiology of heat, body temperature mechanism is hypothermia and hyperthermia, that has to be studied. So this is about nervous system. And the next is physiology of endocrine system. This is the last system which comes under part B. Endocrine glands should be explained with the following points. Whenever you are explaining the endocrine glands, you have to explain the endocrine glands with the following points. First point is functional anatomy of the gland. In that situation, parts, histology, blood supply, and nerve supply should be studied. Second point is functions, which hormone it is secreted and what is the action of that hormone, that has to be explained in the functions. And third one is regulation of glands. In that in that regulation by other glands, which gland this gland is regulated. For example, endocrine glands, which glands, which gland it is regulated, what are the other factors, feedback mechanism, those regulations of these glands should be written. And applied aspects of the particular gland, that should be written, that is hypoactivity and hyperactivity should be written. Whatever the gland may be, you have to write, you have to cover all these points in that gland, then the answer will be very neat. In case of hormone, how to study hormone should be studied under 9 points, that is source of secretion of hormone, chemistry of the hormone, half life of the hormone, synthesis of the hormone, metabolism of the hormone, action, that what it does, and mode of action, how it does, and regulation of the hormone and applied aspects of the hormone like hypo and hyper activity of the hormones. So all these things should be studied under the study of hormone. I will not go in detail about the each and every hormone and each and every endocrine gland, but the each and every endocrine gland studied under the four headings, that is functional anatomy, functions, regulation of gland, applied physiology, and all the glands endocrine hormones are studied under these nine headings, that is source of secretion, chemistry, half life, synthesis, metabolism, action, mode of action, regulation, and applied physiology. So with this I conclude my class of this presentation. uh, Over to you.
Thank you, sir, for the wonderful, knowledgeful, and a beautiful session. Um, shall we move ahead for the Q&A session now, sir?
Yes, yes,
Question and answer session.
yes, yes.
Sir, can I remove this presenting? Oh, okay, sir. You only remove. So I request the viewers to start putting their questions up in the chat box, and I will be reading that out to sir, and sir will be answering your queries regarding Kriya Sharira or regarding exam. You can start posting those questions. So, sir, we have our first question coming up. So the question is, paper one and paper two separately, we should get 50 marks for passing, or by aggregating both papers 100 marks. This is the question, sir.
Okay. uh, Paper 1 and paper 2 should be aggregating. It is not aggregating. You have to get 50 marks in paper 1 and you have to get 50 marks in paper 2. That is the thing. It is not aggregating. In both
Okay,
papers you
sir.
have to get marks, 50 marks.
Okay, sir. Sir, I would just like to add a point. I don't know whether it's completely correct or not, but what we got is, or what we had is in RS6 and RS5 batches, and what the information we got is, few said that it's aggregate of both the papers, and the minimum is 40 in one paper, but aggregate of like 40, 60, 41, 59, like this, it's possible. That's what the information we got, sir.
Right, but it is not for this RS7 batch. This is new syllabus. This is syllabus by NCISM, right? So it is not there for this batch. And in this also, in evaluation you have to, there will be three evaluations, and in that whichever the highest marks will be getting, that will be considered as the final marks that is there for this batch also.
Okay, sir. Thank you for the clarification, sir. So the next question we have is, in ahara parinamakara bhavas, should we correlate to modern?
My point of view is, don't correlate anything which is Ayurvedic topic, which are Ayurveda topics. For example, you have given the example is ahara parinamakara bhava. I wanted to give example is rakta. So what is rakta? How you are going to correlate with modern this thing? By similarities you can say that rakta can be correlated to blood, but not rakta is equals to blood. Rakta is more than blood. Okay, so don't correlate the things. But in while explaining the ahara parinamakara bhavas, you have to uh, uh, ex- you have to explain the significance of those ahara parinamakara bhavas. Okay, significance, why, how ushna is helping, how vayu is helping, how kleda is helping, how sneha is helping. So those significance should be explained here. You can correlate, there is no mistake in correlating, but don't mix and make masala out of it. Okay.
Thank you, sir. Thanks for answering the question. Uh, as we head to our next question, the question is, uh, what are the important shlokas in Avastha?
Avasthapaka, there are three Avasthapakas explained, that is Madhura Avasthapaka, Amla Avasthapaka, and Katu Avasthapaka. For each and every Avasthapaka, there is one shloka, that is for Madhura, Annasya Bhukta Matrasya, for Amla, Parantu Pachamanasya, that shloka is there, for Katu, Pakvashastu Praptasya, that shloka is there. So only those three things shlokas are enough for my perception of.
Thank you,
If you
sir.
write those things, it is enough.
Thank you, sir. So the next question we have is, while writing about any dosha, sthana, and karma, should it be correlated to modern? The question is same
This
again.
is my humble request. Don't correlate any Ayurveda topics to the modern. Okay, if you correlate, you won't end up with the proper answer. Correlation is very good, but don't mix up the Ayurveda points in this paper and the modern points. You can correlate. There is no mistake in correlating. You can say that. um Rakta is blood, but not exactly Rakta is equals to blood. Rakta is more than blood. There are different things are there in the Rakta than the blood. So for that purpose, don't correlate the sthanas of doshas to modern things.
Thank you, sir. Sir, the next question we have is it necessary to memorize shlokas for Manasika Prakriti?
For Manasika Prakriti, uh, if you are what you call for those who can by heart slokas very easily, for such people it is they can by heart slokas, memorize slokas. But if you are feeling difficulty in by hearting slokas, it is okay. For 5 marks, if you explain all those characters, if you remember and explain all those characters, then it is okay. If you write shloka, then it is well and good.
Thank you, sir. Uh, the next question we have is regarding the about shlokas only. That's the student is asking about, is shlokas are necessary for Sama and Nirama dosha?
Sama nirama dosha. That is what, if you can by heart these shlokas, yes, you can by heart. If you write in exams with the shlokas and its explanation, it is well and good, you will get a good number of marks. If you don't write the shloka, if you write only a description or explanation about the particular topic, then you may not get the desired amount of marks or a good number of marks. So better you write the shlokas and its explanation, along with its explanation, it is well and good.
Thank you, sir. The next question we have is there any important topic we need to focus in endocrinology more, sir? It's the topic which has been covered recently for us and it's huge. So anything to focus particularly is the question, sir.
If I start with the endocrine, again it will take another 1.5 and 2 hours. So it is difficult to explain all the things in a single in a short period of time. Here I have clearly mentioned that all the endocrine glands and all the endocrine hormones should be studied with the help of these headings. For example, endocrine gland, for example, pituitary, you take a pituitary gland. What is the structure of pituitary gland? What is the functional anatomy of pituitary gland? What is the situation? Where it is situated? What are the parts of the pituitary gland? Histology of pituitary gland. What is the blood supply, nervous supply? Next is second point is functions. In the such pattern, if you study, if you write in the exams, it is you will get more number of marks. than the regular presentation of glands. It is there in Shambulingam also, and even I follow the same pattern of explaining the gland. And in case of and it is same with the hormone also, every hormone also.
Thank you, sir. The next question is, sir, could you please explain an example for Vadi Kriya Kala?
Example for Vyadi Kriya Kala. Uh, right now, Vyadi Kriya Kala example is you take any Vata Vyadi. With that, you can explain in the beginning. In Chaya, Prakopa, Prashama, in those things, it has to be explained. I don't have proper answer for this right now, because I have not covered that point in this presentation. For exam point of view, you explain each and every Kriya Kala with the example of one disease or one particular thing you take and you explain this. That is the thing.
Thank you, sir. Uh, I think that's all from our question and answer session, sir. And one thing I would like to ask you, sir, on behalf of all the students, if you would allow me to, I would like to
Yes,
ask a question, sir. Thank you,
yes, yes,
sir.
yes.
So, even you may have heard this, sir. Since post COVID times, for whenever an examination is announced, and whenever the examinations are getting near, just like 10 days or 2 weeks, there's this rumor that starts regarding the postponement of exams, which actually disturbs the students.
Yes,
It
yes.
pretty much disturbs the students, and students starts to believe it, and starts to ask different organizations, some influential people on Instagram who claim they will postpone exams. Some Ayurveda pages, they claim they will postpone exams. There's a chances, fair chances. They are this few things, few miscreants who misguide the students, which actually the postponement is not unlikely in this condition. They have announced the timetable well before 45 days, and it's unlikely. There's no uncertainties for postponement. During COVID, there was COVID, so there was a certainty that exams will be postponed. Now there's no uncertainty, and there's no any such incident or circumstance where the university will consider postponing of exams. Even the all tickets has arrived yesterday only. So what would you like to suggest the students regarding this thing, to not to believe in any such things during exams? And also, what would you like to advise the students to face the exams bravely? What are the tips you would like to give them, sir?
Uh, Regarding this point, there are different rumors at the when the exams are nearing to the when the exams are nearer. So don't concentrate on those rumors, because uh, at one stage, during the nearing the exam, your speed of reading and your speed of working out on the subjects will be very fast. Because from long at least 3 4 months you have started the uh, preparation of exam. So this is the peak time, and where your study speed will be very high. And in that crucial time, if you hear such things, the it is human mind, it is human, we all are humans, and it is human tendency that exams are anyhow exams are getting postponed, let us slow down my study. That will be the human tendency, but you should not do that. If you get a if anything happens and exam is postponed, you will be getting the extra days. Those are grace time, those are grace days. So those days can be utilized for any left out topics. So don't concentrate on, don't look for those rumors and spoil your study.
Thank you, sir. Thanks for the suggestion and advice that you gave. So there's one more question which just popped up. Uh, Sir, is Prakriti topic important as we have it for practicals too?
Yes, yes, yes. It is very important because uh, in Prakriti, we can expect the uh, 10 marks question and 5 marks question also. So it is must and should, and it is very important topic. And if you study here, it will come in practical also. So in one shot, you will be you will be achieving two goals, right? So so read here only, and by heart all the shlokas of Prakriti, and read in the this topic only, this paper one only, so that it can be utilized for practical also. In practical, it becomes very easy, right? So read, it is very important topic.
Thank, thank you, sir. I think now that's all from our question and answer session. Thank you for clearing the doubts of the students, sir. Uh, I hope they were understood and found answers for their queries. So, coming to the end of the session, as we proceed to the end of the session, I would like to thank on behalf of my whole organization and team at Jignasa for accepting the invite to deliver a guest lecture series on Kriya Sharira, sir. Delivering a lecture series on a specific topic is different. Delivering a lecture series on whole paper is
Also,
different. Yeah, sir, whole subject is different. And that was a, I think it was a challenging task, but you accepted it, sir. Thanks for that. We are really glad for that. We arranged topic wise sessions on anatomy, but due to
yes,
time constraints we couldn't do the similar for other subjects. So we had a challenge on arranging a session on for a whole subject, and you got on board with us for that, and you delivered it, sir. So it's really overwhelming for us, that what we thought of, you got it delivered, sir. So we are really grateful. We are really thankful for that, sir. And in future also, we look forward to collaborate with you. We look forward to work for the Ayurveda fraternity, Ayurveda students with you, sir. And once again, we thank you for all the efforts you have put in delivering the session today, sir.
thank you for calling me, and thank you, Karthik, and thank you, Jignasa team. Thanks for giving me an opportunity. Thank you, thank you.
Thank you, sir. I request your permission to end the broadcast now, sir.
Yes, and
Thank you, sir.